Hello, thank you for listening to my presentation today. I'm eager to talk about the research we've done on our specific strain of Bacillus vallis mortis that we've isolated from the bank of the Great Salt Lake, and the interesting study we've looked at to see if it's able to prevent or isolate or resist fungal growth in um, environments resembling the human urinary tract. This idea was grown out of a study that we discovered Hoppy et al. That was performed in the late 90s, where a similar bacillus species was administered to rats who had recurring urinary tract infections. And so um, antibiotics were administered, and then they also added this bacillus species to that environment to act as a potential competitive inhibitor. They showed in that study that it did work, that administering and applying a new microbiota was able to prevent the recurrence and recolonization of the area by the pathogens. And so we are looking into a similar study. We isolated this Bacillus vallis mortis from the soil of the Great Salt Lake. Um, we found that you can see here that we found that it was a bacillus, that it was gram negative. Using PCR, we were able to identify it as a strain of Bacillus vallis mortis. And in the research we were doing, we also found that Bacillus vallis mortis is used as an antifungal in agricultural capacities for plants. And so as we found this study, we wanted to see if we could resemble that study with our Bacillus species. So as you can see, here are some of our identifying studies. We measured growth via absorbance, light absorbance in broth at different temperatures. And as you can see, there's an increasing trend up to 40 degrees Celsius, which is approximately human body temperature, internal temperature. And so, we took that detail. We knew that it would grow effectively. It would survive at the temperature. We also needed to know if it would survive um, in the specific chemical environment. We knew because it came from the Great Salt Lake that it could survive in salty environments, but we had to test if it could survive urea. That's what this figure here shows. These plates are made with an indicator. The red shows that it's processing urea, and that they were inundated with urea. So they started off yellow, and as the bacteria grew and processed urea, it became this pinkish red, more acidic um, result. So we proved a couple of these results back to back. We knew that it could grow in the and we knew that it could grow in the urinary tract, hypothetically. Of course, we didn't test in vivo here. Um, and so the next thing we did is we started applying fungi, different types of fungi, to these plates to see if we could observe zones of inhibition. This is where we ran into some issues with our study. We found that our fungus really did not like growing in these urea-rich, salty environments. And so we tried several species, didn't see much growth. And when we did see some growth, it was very splotchy and did not seem to interact with our bacteria or with the E. coli control. So it's an important takeaway here that we did not necessarily find support for our original question as to whether or not this could competitively exclude um, fungal pathogens from the urinary tract. But we did find that it will grow, and it's well documented that Bacillus vallis mortis is antifungal. It can inhibit fungal growth, perhaps just not our strand. So in terms of following up on that, I want to draw your attention here to figure two. Um, we observed growth size of our different treatments. We made several plates using these urea growths, um, and each plate was split down the middle with our isolate on one side and with our E. coli control on the other. The isolate growth, average colony size, is in blue, and the average E. coli colony size is in orange. And you can see at every plate, it is not just larger, it is significantly larger for our isolate. And so there's further research that needs to be done to see if this is potentially useful as a fungal inhibitor in animal trials, but that is something beyond our reach. Um, but we are very pleased to show that it did in fact grow in these environments, which is perhaps the first step to looking into it as an antifungal inhibitor further down the road. So thank you for listening to my presentation, and I hope if you have any questions, you can reach out to me.